Hello, and welcome to today's webinar. I'm Theresa Crimmins. I'm the director for the USA National Phenology Network, and I'm so excited to be sharing with you some of our more recent uses of the data that are contributed to Nature's Notebook. So today, I am so excited to be sharing with you some of those recent ways that the data have been contributed. This is a part of our 15-year celebration already for Nature's Notebook. This week, we are celebrating Phenology Week, and a key feature of the week is that 2023, this year that we are in, is the 15th year that Nature's Notebook has been running. And so we've got all sorts of really excellent things planned, including a party, I think on Monday, and also an event on Friday of this week. Please check out USANPN.org/Phenology Week 2023 to learn about all the opportunities and events. And I know good stuff is going to continue to be added to that website as we get things finalized. So as always, I want to start by saying thank you. Thank you so much to everyone who has supported Nature's Notebook in all the time that we have been in existence. Thank you if you contribute phenology observations, even if it's only been one. Every single data point matters. And thank you for being a supporter of the organization and the work that we do. It truly is a critically important data resource that is used increasingly more by scientists to better understand how our ecosystems and communities are being impacted by changing climate conditions and other pressures like fragmentation and development. And the information is used increasingly more too to support a whole wide range of decisions, anywhere from anticipating whether the wildfire season is likely to be a bad one, or when to take action to manage invasive species. And so I'm going to share just a couple of the most recent uses of these data with you today, but we have ample opportunity to learn more if you are so inclined, and I'll cover some of that at the end. So just a quick refresher. I go over this every time, but I'm sure it's helpful. The reason why the USA National Phenology Network was created, and the primary thing that we do, is to collect, store, and share data and information about when plants and animals undergo seasonal transitions. Why do we do this? Because these data and this information is so critically valuable for advancing our understanding of these species and what they're doing and how they're changing, for informing a whole wide range of decisions, for better communicating and connecting. And our final activity is to create and grow an equitable and inclusive network. So some quick summaries of where we are at and what we have achieved in the past 15 years. This graph shows our annual accumulation of records coming into Nature's Notebook, starting at the bottom with the red line representing 2009. The x-axis here is the day of year, and the y-axis is the total number of records. And I think we achieved a couple 100,000 records total in 2009. And then we go basically in rainbow order for many years in a row here. And you can see that in every subsequent year, we accumulated more records. And that pattern continued all the way up until 2020, which is this black line here, which was COVID. And that did put a bit of a damper on the activity, although it really wasn't that bad, honestly. These 3 years here, the brown and the navy and the purple, were 2017, 18, and 19. And they were all pretty much right there, pegged at about just over 3.5 million records in each year contributed. And then the gray line up here is 2021. You'll notice that 2022, which is the pink line, is a bit below those other lines. And that is because we have a big chunk of data that hasn't yet been ingested into our database. We've been setting that activity aside in favor of trying to get our new website finalized and launched, which is set to occur in a matter of weeks. And then after that, we will make that final big data ingestion. And I know that that pink line will pop above every other line on this graph, showing yet again that we have set records and brought in more data than in every previous year. And then just as a little status check, this kind of fuchsia line that we see pretty much on top of every other line is so far where we're at in 2023. So again, thank you to everybody on here who has helped contribute to that line, because that every single data record that you contribute matters, and you can see it captured here. When we take that information and turn it into a map, we can see where are those data coming from. And if you stare at this long enough, you might see your house or your site where you monitor represented on this map. Every site that data have come into in Nature's Notebook is reflected on this map. And then the size and the color of the circle reflects how many records have been contributed. And what's really cool is that whenever I update this map, which is usually at best once a year, I always have to adjust the legend to accommodate sites with more and more records than we've ever had before. So that is always very gratifying. And again, thank you. And then finally, this map indicates where we have our local phenology projects. The black dots are sites where individual observers have been contributing data, and thank you to all of you for that. And then the green dots are sites managed or where local phenology projects have been established and data have been coming in. And every time I update this map, I'm very excited because the number of green dots in particular always shows significant growth. So how are the data that are contributed to this program used? I would love to answer that question for you. Today, I'm going to cover just a couple of interesting examples. We are now over the 130 mark for peer-reviewed scientific papers that have used the data in one way or another. And that doesn't count all of the ways that the data are referenced in a more anecdotal or casual way, or where maps are simply consulted by decision makers. It's not even possible for us to track all of that. But when we do hear of folks using the information to support, say, decisions on when to go out and treat for invasive species, or even when would be a good time to film for a movie or a TV show based on the tree canopy developmental status, which is something that we've answered questions on in the past. We do keep track of that just in a qualitative way, because it's really helpful and interesting to us. But today I'm going to cover 3 of the most recent papers that have been published, and they represent 3 different ways that the data have been used. One is in better understanding what are the conditions that seem to drive activity in a particular group of species. Another is in better understanding and being able to predict when events of particular concern are likely to occur. And then the final is in looking at comparing the ground observations that are contributed to Nature's Notebook to data that have been collected by a satellite, so something that is commonly referred to as remote sensing. The first one I'm going to talk about is I have a little calendar icon here because it is the example, it's an example of where being able to better anticipate when something is going to occur is meaningful and helpful. And in this case, it has to do with something that probably a lot of you are really intimately familiar with, especially this year. If you're in the East, spring got off to such an early start, and I have been hearing over and over and over again how the allergy season just really ramped up early and is really intense so far this year. We would love to be able to answer that question, the question with more confidence: when will pollen be most abundant in the air? And so we are actively trying to investigate whether the data that are contributed to Nature's Notebook can help with predicting when allergies are going to be bad. So I was surprised to learn, and you might be too, you might be thinking, but there are forecasts of pollen in the newspaper and on the news and in the daily forecasts that I watch. It turns out that for the most part, those predictions are not based on actual observations of pollen. In a lot of cases, those forecasts are based on things like when and where allergy medicine has been sold, or other interesting proxy variables. But in this country, we are fairly limited in our actual on-the-ground monitoring of airborne pollen. And so it does limit our ability to be able to better predict when pollen is going to ramp up. What we do have is a network of about 80 active stations where folks install counters or sensors like this one that we see here. This gentleman's up on the roof of a hospital. And these sensors are passive and they are collecting samples of what's in the air over time. And what it requires is that an individual has to go up, and it's usually daily, and take a sample that was collected by that piece of equipment and then bring it down, and somebody who is trained has to look at the pollen that was collected through a microscope and identify the individual grains and then try to estimate the quantity. And so, as you might anticipate, this is a rather labor-intensive process. In addition, these stations are, for the most part, privately run, either by hospitals or by individual allergists' offices. And so the data aren't that readily available for the most part. And so what that leaves is a really big gap in where pollen is, where and when. We just don't really have very good information about that across our country. And so it seemed to some researchers that the flowering information that's coming into Nature's Notebook could be perhaps a piece of that puzzle. Because in truth, you can't have pollen flying in the air unless you've got flowers that are open, releasing that pollen. And if you're a part of Nature's Notebook, you're probably keeping track of when you're seeing open flowers on the plants that you're observing. And so what we endeavored to do was try to see if we could establish some relationships between what has been contributed to Nature's Notebook and the observations that have been documented at those stations in that map that I showed on this previous slide. These are actually stations run by an organization called the National Allergy Bureau. And so what we did was requested the data set from the National Allergy Bureau stations, and the locations of those stations are shown on this map as the larger black dots. What the National Allergy Bureau station data look like are counts of pollen grains observed on a day at a location, broken down by taxa. Typically that taxa is at the genus level, so that means either maples or oaks or alders or what's another good example? Ragweed. And then in a few cases, it's actually at an even higher taxonomic level, the family, in the case of Cupressaceae, which is the family that junipers and related plants are in, or pines. They don't distinguish among the pines, it's just the Pinaceae family. But what we opted to do was look at the flowering observations that have been contributed to Nature's Notebook in a region surrounding those NAB pollen monitoring stations and see if there was much of a relationship. And in this first analysis, what we did, we focused on oaks, and specifically, of course, flowering observations for the years 2009 to 2021. And we focused on the eastern half of the United States, this region shaded in the darker gray. The reason being that once you drift into the west, you potentially have plant flowering activity that's influenced more by moisture. And we wanted to keep it simple in this analysis. We wanted to really focus on locations where we thought springtime temperature was probably the most influential variable on triggering or indicating when oak pollen would be airborne. So that gave us, we were working with over 200,000 observations of oak flowering. And so if you observed flowering in any oaks during that time period in the eastern half of the US, I can guarantee that your data were used in this analysis. So thank you. And so here's an example of what oak flowers look like. Those ones aren't yet releasing pollen, but they look like they're getting close. And this is the kind of thing, this is the exact kind of observation that is so critically valuable for what we're trying to do. So what we aimed to do in this study was identify whether there was a relationship between when oaks were observed in flower, which is on the y-axis here, and the preceding winter to spring temperature. And what we found was, yes, there actually was a pretty strong relationship there. The R squared here is 0.66, and that's reflecting that there is a pretty significant relationship there. Well, there's a pretty strong relationship, and then this p-value indicates that yes, indeed, it's very statistically significant. And so what that is showing is that we see earlier flowering when oaks are observed, when they're exposed to warmer temperatures in the winter and spring months. And specifically, for every degree warmer that a spring is, or a location is, flowering advances in oaks by 3.5 days. And so again, if you're in the eastern United States this year, you are witnessing this in real time yourself, because it has been so much warmer than average, and those plants are flowering and releasing their pollen much earlier than average. So that's all well and good. It honestly wasn't surprising to us because we know this. There's been lots and lots of studies that have documented that, again, especially in the eastern US, springtime plant activity is by and large governed by temperature. And it really has a lot to do with winter and then especially spring temperatures. So we expected to see a strong relationship there where warmer temperatures would lead to earlier flowering. But what really matters is we also wanted to see, was the timing of flowering as reported by Nature's Notebook observers indicative of when pollen would be peaking in the same locations. And that's what this graph is showing. The day of year for the peak flowering date, and it says modeled here because we had to interpolate a little bit with the data, is on the x-axis, and then the date that the pollen peaked is on the y-axis. And again, there's some variability here around this line, but generally it's a really quite strong relationship. And so what that's telling us is then, yes, flowering in oaks is driven by temperature, and we know that it's warmer. When it's warmer, then it's going to be earlier. Also, flowering is tightly tied to when pollen is in the air. And so therefore we can step back and take the flowering observations actually out of the equation and say that temperature should be able to help us predict when peak pollen will be occurring. And so this is really, the intent here was simply to be a demonstration of a test and a demonstration of the ability of Nature's Notebook flowering observations to support pollen forecasting, but it performed really well. And we were pretty excited to see this. And so we now have quite a bit of additional work taking place where we're going a lot further to try to extend this to other taxonomic groups and develop much more complex and sophisticated models too, with the hope that someday the National Phenology Network would actually have pollen forecasts. And specifically, and what is unique about what we're trying to do here compared to what you might see on the Weather Channel or in the newspaper right now, is that we are addressing it from at the level of the different plant taxa. You might notice that when you see a pollen forecast or a pollen indicator on the weather station, it usually is grasses, trees, molds, and maybe one other thing. What we're actually doing is trying to develop those relationships at that taxonomic level of oaks or maples or alders or ragweed. And the reason why that matters is that different individuals are sensitive to different taxa, and it makes sense to be able to tailor when you take your medication based on when those taxa that you're sensitive to are likely to be most abundant in the air. And so that's an exciting and novel thing that we hope will emerge from this work eventually. And so to support this work, we've launched a new campaign this spring that is nationwide in scope, where we're asking folks to observe when they see flowers open or closed, because that matters just as much, in all of these different plant taxa, because all of these are plant taxa that are observed or are being tracked by the National Allergy Bureau, by folks that are looking through their microscopes and counting pollen grains. And so our hope is to eventually be able to establish what the relationships are between flowering and pollen dynamics using these kinds of observations. And so we invite you, if you're so inclined, to please join this campaign and tell us when you see these different species of plants in flower or not in flower, because again, that really matters too. Okay, shifting gears a little. The next study that I'm going to describe is one where the researchers were focusing more on what are the conditions that seem to drive particular events. Like I said in the previous example, some things we know fairly well, like for example, we generally know as a broad brush that springtime activity in the east, and especially, is driven primarily by temperature. But when it comes down to the individual species, or even in different locations, we don't always know the specific drivers. Sometimes it is just temperature, but sometimes it also has to do with something like snow melt or day length, or even sea ice, depending on if we're talking about an aquatic species. And so in this particular study, the researchers were focusing on different functional groups. In this particular study, which focused on the northeastern US, researchers were looking at what seems to drive leaf out in trees, woody species, versus perennial herbs, the stuff that we see growing on the forest floor. When we back out to the broad brush again and what we know generally, it seems that in temperate systems like the eastern US, primarily woody species seem to be driven by air temperature. And so again, like we're seeing in the spring, it's been warm, air temperature has been warm in the east, and so we have early leaf out in a lot of those woody species. The perennial herbs, a little bit different because they care a bit more about soil temperature and snow depth. And so if we have more snow, it can insulate them more and actually change the timing of when these things leaf out compared to the overstory canopy trees. What we do know is that when we have different cues driving activity in different species or different groups of species, that can lead to, over time, as conditions change, things that historically occurred at the same time might start to drift apart. We don't actually know what to expect out of this. If understory canopy forbs or herbaceous plants are cued by soil temperature and snow depth, and overstory trees are cued by temperature, it could go either way depending on how things change. We might see the understory herbs advancing their phenology more and becoming active earlier in the year, or we might see the opposite, where the trees advance their phenology more and become active earlier in the year. We don't really know, but either change stands to have impacts on things like carbon cycling and how successful the different plants are reproductively going into the future. And so a group of researchers opted to try to answer this question as to how things might be changing among these two functional groups using observations of all these species, overstory trees and understory herbs, in this whole big region. It ended up being, and it was all data contributed to Nature's Notebook. They pulled data from 965 sites using data from 2009 to 2021, 10 species of trees and 11 species of herbs, and it ended up being over 8,000 observations. So if you squint at that map, if you're up in that region, you might see your site. And again, if so, thank you so much. What the researchers asked here was, do the deciduous trees and the understory forbs show different responses to temperature? And if so, what might that mean for how they change going into the future? And then they also asked, does this vary by latitude? There's the slide I was looking for. I whizzed right past it. So because they broke it up by latitude, we have three different plots here. I'll start with this one here because this is the lowest elevation in the area, in their study area. And it's broken up into three elevation bands. This is the lowest, this is the medium, and this is the highest. And the dots again are shaded by whether they were observations from herbs or from trees. And then there's the line of best fit through each of those, and it's shaded with the same color. And so what we see here in all three of these graphs, the mean spring temperature is across the x-axis, and the day of year of leaf out is on the y-axis. And so as we saw in the previous example with the oaks, when it is a warmer spring, things happen earlier. That's basically what these lines are showing. And when we look at this lowest elevation graph, those lines are effectively parallel for those two different functional types. And so both functional types, both trees and herbs at low elevation, are showing the same thing. And they are, what this suggests is that if temperature conditions were to change, they probably would advance their phenologies together, and things wouldn't really be too impacted among these two functional groups in that area. And the same is true at the middle elevation. But once we get up to the upper elevation, we actually see really different responses among the two groups. And in particular, the herbs are advancing their phenology at a much greater rate as compared to the trees with the warmer temperatures. And so this kind of differential change between these two functional groups could have some significant consequences. What can happen here when we have an earlier leaf out or longer growing season in the trees, one of the things we might see with that is that the trees, when they put on their leaves earlier, can actually take up more carbon from the atmosphere and store it in their plant tissues, which is a good thing. However, some studies have suggested that when plants get going earlier in the season like that, they kind of tire themselves out, and by the end of the season, they need to drop their leaves earlier than they would. And so the growing season length doesn't actually lengthen, doesn't actually get longer, it just shifts. And if that's true, then that earlier season carbon uptake is canceled out by the tree dropping its leaves earlier as well. If the spring ephemerals are putting out, or the forbs, the understory forbs are putting out their leaves earlier, that could put them at greater risk for frost damage. That's definitely a situation that we have contended with and may continue to contend with this spring, again, where we have such early activity. And it's not really spring yet. There's still definitely the potential for freezing temperatures and snow and ice in a lot of locations where plants have started to put on leaves and flowers. And that could become more of a consequence going forward. And finally, the differential changes in these two different groups, these plant types at the highest elevations, could also have implications for pollinators, depending on whether pollinators shift their timing or not, and whether plant or animals that eat the flowers or the leaves change their timing or not. It could really either release pressure on plants, on particular species, allowing them to flourish more, or it could put additional pressure on them and create additional stress. A lot of it is not clear at all, necessitating further studies, further pointing to the value of the data and observations that you're collecting. But this finding here alone is novel and is truly an excellent example of the kinds of findings that are made possible when we have data across a large geographic region, like we have through Nature's Notebook. Prior to this study, there had been a couple of other examples where researchers had looked at different rates of change or sensitivity to conditions among different functional types, but they were site specific. And what's very unique about this study, what sets it apart, is the fact that it is across that whole large region of the Northeast, and that only is made possible because we have observations from so many sites coming in through Nature's Notebook. So very cool, and very much appreciated that these data are continuing to come in. Oops, I already talked about that. Okay, and then shifting gears one more time, the final study that I would like to describe is comparing, showing how ground observations can be used in comparing or an analysis of imagery that's being collected by sensors mounted on either drones or planes or satellites. we keep seeing more and more and more of these sensors being launched and increasingly high resolution data being available from these sensors. However, what we're seeing, and I think will become apparent in this example, is there's still no substitute for observations on the ground. And the imagery collected by these sensors still requires some kind of ground validation, some kind of eyes on the ground to be able to connect the dots and say, yes, that thing that you're seeing in that imagery indeed is this thing that I see on the ground. And this is one of the really the many examples of where ground observations collected through Nature's Notebook play that very critical role. I think we've got, we're probably close to 15 or 20 studies, maybe even more now, that have leaned heavily on observations contributed to Nature's Notebook to help validate and verify what's on the ground when folks are using imagery collected from remote sensors. Okay, this is a very complicated diagram, but I like it a lot because it shows how, as conditions change on a tree here in this example, there are both drivers and responses. And so this is one of the main reasons why phenology matters so much. It both helps shape the environment and is shaped by the environment. And so if we start at the top here, which is basically the season that we're in right now, we've got days starting to get longer, temperatures starting to get warmer, trees put on its leaves, that's the leaf flush part. And then we move on to the flowering. And then once we've got flowering, we've got nectar feeding, pollination, the tree starts to take up more water. Then we move on to fruiting. We've got interactions with other species who depend on those fruits as a food. We move on to seeding. Well, seeding and fruiting are the same thing. It depends on what the species is. We continue to have soil water uptake, but it decreases as we start to move into the fall. We start to see leaves come off as the days get shorter. And what's even more interesting here is that as these things are all happening, the environment itself is being impacted too. We have things like when the leaves come out, the interaction with at the local level in trees make oxygen for us, as we know. They take up carbon dioxide, they let go of oxygen, and we start to see different changes in the very local atmosphere of the plants, and then that can start to have impact on boundary layer conditions and air surface patterns and all sorts of other ways too. And so phenology is really important for its governing nutrient and water cycling as well as species interactions showed in this complex diagram. However, that's a generalized statement. And again, there's so much variability from one species to the next in terms of when these things happen, what kind of influence they have on their environment, what the ways in which the environment affects them. And actually, it even varies a whole lot from plant to plant, which argues for taking observations on a whole lot of individuals if you really want to be able to understand what are the conditions that drive, say, leaf out or flowering, or the ways in which the plants may be impacted by local conditions. And so we're doing a fantastic job with that in Nature's Notebook, where folks are observing thousands and thousands of plants. But we actually need so many more observations if we really, really, really want to know what's going on. And so one of the ways we could potentially increase our sample size and have information coming in from a lot more individual plants is to take advantage of imagery that is collected by sensors that are remotely collecting data and combine that with those observations that we are collecting on the ground through Nature's Notebook. So remote sensing 101, real fast. What I'm talking about, here's an example of what we're talking about. This is a satellite, and as you probably know, the kinds that are collecting the information that we use here are orbiting the Earth and constantly sending down signals and having those bounce back to them, and they are recording what is bounced back to them. And basically it is the amount of energy reflected in different spectral bands or in different segments of the visible light spectrum, as well as infrared, near infrared, ultraviolet. Oh, so wavelengths that our eyes can't see as well or at all also. And all of that information is then pinged down to storage facilities on the Earth, and that information can then be processed to better understand what might be happening on the Earth. Whoops, there are a whole lot of those kinds of satellites, like I said, and they all collect data at different spatial resolutions. And what's cool is that it seems like just about every day, more satellites are being launched that are collecting data at extremely high resolution. And so in this study, the researchers opted to look at a data source called PlanetScope, which is 3-meter resolution, I believe, which definitely allows you to identify distinct trees or tree canopies, as we could see in these images, and compare them to the observations being contributed through Nature's Notebook to see how well the imagery seems to capture and tell us when leaf out or leaf out is occurring, and then compare that to what we know observers are seeing on the ground to see if the algorithms that process the satellite imagery do a decent job. And so what we are looking at here are 3 different sites that are under constant, very repeated, very regular observation through Nature's Notebook. They are part of a network called the National Ecological Observatory Network, or NEON. That network is funded by the National Science Foundation, and they are sites located all across the US where a whole lot of biological and abiotic parameters are collected, and the data are made freely available for scientific discovery. What's really cool is that NEON uses the NPN protocols, and they've got staff that go out at least once a week, I believe, during the active growing season, and then every other week at other times of the year. And they use the exact same protocols that you do to track what they see happening on plants at these facilities. And so the dots that you see in these images are individual trees that are under observation by the NEON technicians at 3 different sites. A is Great Smoky Mountains, and then B and C are Smithsonian Institute facilities. These are all in the eastern United States, and you could probably guess that because those are nice eastern deciduous forests. And the different color dots represent different species. And so if you're a Nature's Notebook observer, this will look very familiar to you. This is the general protocol that you answer questions on when it comes to deciduous trees. You are asked about the status of breaking leaf buds, increasing leaf size, leaves, and colored leaves. And if you say yes to those at any given time, you are then prompted with how many or how much of those things do you see? And so again, the NEON technicians are using that exact same protocol. And so we have regular observations for all of these phenophases for all the trees that are shaded here, as you see. And so those are the data that were treated as the correct answer in this analysis. And the imagery-based information, it was compared to that to see how well it performed. And so these are the data from one of the years from the Great Smoky Mountains for, I forget which species, but this is one of the species. And so what we see in the orange dots is, I think that one makes more sense to look at first. The x-axis here is the day of year. And then what is shown in orange, and it's reflected on the y-axis over here, is the leaves intensity. And so they took that information from the protocols and converted it to values that they scaled from 0 to 6. But basically what this curve is reflecting is when do we see no leaves, and then when do we start to see those leaves, when do we have max leaves, and then when do we start to see those leaves drop back off. So that is a depiction of the reports coming in through Nature's Notebook. And then the blue dots, what those reflect are effectively something called greenness. These are, the images are brought in and then processed using an algorithm. And in this case, they use the extended vegetation index. I think that's what EVI stands for. Or sorry, it's enhanced vegetation index. And it has to do with a differencing of the red band and the green band and the near infrared band. But basically, it's an algorithm that performs really quite well for estimating how much green is there in this particular element of the image that we're looking at. And the amount of greenness should reflect canopy development, which should be captured well in the Nature's Notebook observations. And so when we look at those blue dots, those are reflecting what we see as pretty low greenness early in the year, and then we see a quick spike, and then it peaks around day 150, 180 here. Then you start to see it drop off, and then a gradual, and then a rapid decrease toward the end of the year. And so what the researchers see here is that the greenness actually does really a quite fine job of capturing the leaf on and the increase in leaf size, and then the progression toward full canopy. But then it does kind of falter a bit as you get later in the season. And the reason for that is that as leaves age, they lose some of their chlorophyll. And so the image doesn't reflect as much, the trees don't reflect as much green. And so the image that the sensor is capturing and then the algorithm processes says you're losing green. Whereas an observer says, I still see a full canopy, and your eyes actually can't detect that degradation in the chlorophyll. And so we see a nice flat line in terms of a full canopy all the way up until when the leaves start actually changing color and dropping off. And so the imagery doesn't do quite as good of a job at capturing fall and when leaf color starts to change or when leaves start to drop. They really don't converge again until it's back down to zero. And when we look across the sites, there was a really consistent pattern of that phenomenon. And what was interesting is that even at one of the sites, the greenness never even got that high. This is probably a site that isn't deciduous forest. And so what this study demonstrated was, yes, indeed, remote sensing can help us understand dynamics of leaf canopies over the course of the season. It can definitely help us get pretty good estimates of when that spring leaf on occurs. And it might do okay in terms of capturing senescence and the end of season information too, but it isn't perfect. And it may definitely allow us to monitor more trees than individuals might choose to do. However, the truth is there's probably still no substitute for those actual observations that you're collecting on the ground. We still need them. We definitely still need them. And so we're going to keep at it. Okay, are you interested in learning more? Because we try to summarize every paper that uses our data. I hope someday we have so many studies happening that it's impossible to do this, but for right now, we're able to more or less keep the pace. And so if you're interested, we cover them in these webinars that we do once or twice a year. And then we also do these highlighted publications, which you can access on the Nature's Notebook website. And if you receive our newsletters, you'll probably see that whenever we generate a new one of these, we link it in there. But if you don't yet get our newsletters, definitely sign up so you can get access to this information. If you click on that highlighted publications, this is where it takes you on our website, and it just shows you, you'll get a little snippet that summarizes the study very quickly. And then if you click on read more, it gives you a bit more information. Here's what an expanded summary looks like. So what we try to do is present the finding in clear language and explain why it matters and how this contributes to our understanding. And so again, if you don't yet get our newsletters, please be sure to sign up for them, because we always put our best information in there, as well as invitations for new opportunities, like the 2 new campaigns that we are launching this year. The pollen trackers that I mentioned earlier, which is available for monitoring all across the US, and then the Desert Refuge campaign, which is focused specifically in Arizona, with a focus on monarch butterflies and milkweed, which is so important for such an important food source for them. You can also follow us on social media if you're interested in keeping track of what's going on. We're on Twitter, we're on Facebook, and we are recently on Instagram. So come check it out. And with that, I just want to say again, thank you so much. Thanks for your time today, and thank you for your support and your contributions. They are always appreciated. And if you have any questions, please do not hesitate to reach out at any time. Have a great day.